Hello and welcome back to the Biochemistry for Health Sciences channel. We continue our series Understand Not Memorize Metabolism, and today we'll talk about the Kreb cycle of Main Street. In the previous videos, we explained why the generation of ATP from glucose, what we call as Main Street, is probably the most important metabolic highway for the human body. This Main Street is made of 4 phases: glycolysis, PDH, the Kreb cycle, and the electron transport chain coupled to oxidative phosphorylation. That's our Main Street. So we did talk about glycolysis in the previous video, PDH. So today we will focus on the Kreb cycle. So what are the most important features of the Kreb cycle? So the Kreb cycle is made up of 8 reactions. So although we have 9 here, reaction 2 and 3 these are catalyzed by the same enzyme. This is just an intermediate. So basically there are 8 reactions in the Kreb cycle. When you look at the net equation of the Kreb cycle, you will see clearly what is happening in this Kreb cycle. So basically, acetyl-CoA carbons in the form of acetyl-CoA are entering the Kreb cycle, and these carbons are oxidized to carbon dioxide. So you see carbon dioxide coming out here, carbon dioxide coming out here. So basically acetyl, there are 2 carbons for acetyl, and those 2 carbons eventually get oxidized to carbon dioxide. And we can also see that the products of the Kreb cycle include energy, direct production of energy, because GTP is the same as ATP, and indirectly in the form of NADH and FADH2, which will enter the electron transport chain oxidative phosphorylation system, which we'll talk about in a few seconds. So let's look at these 8 reactions of the Kreb cycle and try to explain the most important aspects of each reaction when it comes down to understanding human biochemistry and how we apply it for human physiology as well as pathology. So the first reaction here is a reaction between the acetyl-CoA, which is coming into the cycle, and one of the intermediates of the cycle, oxaloacetate. So basically oxaloacetate reacts with acetyl-CoA and we get citric acid or citrate and coenzyme A. The enzyme that catalyzes this reaction is called citrate synthase. Citrate synthase is belongs to the family of enzymes called transferases. Recall from the previous videos that a transferase is basically an enzyme that catalyzes the transfer of some group from one substrate to another. So in case of citrate synthase, this enzyme catalyzes the transfer of an acetyl group. So basically this acetyl group is transferred to oxaloacetate. Now oxaloacetate is an alpha keto acid. So you see the acid and then alpha to that is a ketone. So oxaloacetate is an example of an alpha keto acid. So citrate synthase basically transfers an acetyl group from acetyl-CoA to oxaloacetate, and what is formed is citric acid or citrate. Since citric acid or citrate is formed, the cycle is also known as the citric acid cycle or the citrate cycle. Now if you look at citric acid, it has 3 carboxyl groups, 1, 2, 3. That makes this a tricarboxylic acid. So the Kreb cycle is also known as the tricarboxylic acid cycle. Okay, so Kreb cycle, citric acid cycle, TCA cycle, they are all different names for the same cycle. This reaction also releases coenzyme A. Now remember, coenzyme A comes from a vitamin, and so this coenzyme A can later be used in this reaction number 5. The citric acid is then converted into isocitrate, and the enzyme that catalyzes this reaction is called aconitase. Aconitase belongs to the lyase family. So the enzyme aconitase catalyzes an elimination reaction. Water is removed, a double bond is formed, and then in the next part, the second part, water is added. There's an addition reaction, and we get isocitrate. So basically, this OH group here of citric acid has moved from this carbon here to this carbon in isocitrate. In reaction number 4, the isocitrate is oxidized to alpha-ketoglutarate, and the enzyme that catalyzes this is called isocitrate dehydrogenase. Remember, dehydrogenase belongs to the oxidoreductase family of enzymes, and these enzymes catalyze redox reactions. So the enzyme isocitrate dehydrogenase uses NAD as an oxidizing agent. So you see NAD here is used as an oxidizing agent to oxidize the hydroxyl group in isocitrate into the ketone group in alpha-ketoglutarate. So once the alcohol group is oxidized to a ketone, now we have this carboxyl group. You can see separated by one carbon, there's alpha, and then the next one is the beta. So this carboxyl group is beta to the ketone. This carboxyl group will be lost as carbon dioxide. And you can see that that carboxyl group has been lost as carbon dioxide in alpha-ketoglutarate. So to remove a carboxyl group beta to a ketone does not require, that's easier to do than to remove a carboxyl group alpha to the ketone, as we saw in PDH reaction. So this reaction does not need the help of a coenzyme TPP. So removal of carboxyl groups in alpha keto acids require TPP, as in the case of pyruvate dehydrogenase, but in this case, this is a beta keto acid. So removal of the acid from a beta keto acid, we do not need TPP. So this enzyme does not require TPP. And also keep in mind this reaction, the NAD gets converted to NADH. So here we are talking about getting energy as this NADH goes into the electron transport chain, oxidative phosphorylation system. In the next reaction, alpha-ketoglutarate is converted to succinyl-CoA. This enzyme that catalyzes this reaction is called alpha-ketoglutarate dehydrogenase. It's an oxidoreductase. Now this enzyme, alpha-ketoglutarate dehydrogenase, is a multi-enzyme complex. It is very similar to the PDH complex. Remember, in the PDH complex, this would be pyruvate, which is an alpha keto acid. So here too, this is an alpha keto acid, and this would be acetyl-CoA in case of PDH. So now we have alpha-ketoglutarate and alpha-keto acid that is going through oxidative decarboxylation, forming succinyl-CoA. So just like the PDH complex, this enzyme requires coenzyme A, TPP, niacin, and so on, similar to the PDH complex. So the product of this reaction is succinyl-CoA. This is a very unstable an unstable chemical. That means it has a lot of energy available in it. In the next reaction, reaction number 6, this unstable chemical succinyl-CoA is converted into a very stable chemical succinate. So the energy that is released by converting a high energy unstable chemical into a low energy stable chemical, this energy is used by this enzyme to react GDP and PI to form GTP. GTP in terms of energy is equivalent to ATP. So this reaction number 6 here is another example of substrate level phosphorylation, something we saw in glycolysis, the formation of GTP or ATP by using energy from a highly unstable chemical. The enzyme that catalyzes this reaction is succinyl-CoA synthetase. This enzyme belongs to a family called ligase. So when the succinyl-CoA is converted to succinate, the CoA part, the coenzyme A, so this coenzyme A will also come out as a product. So as far as the coenzyme A is concerned, we brought in a coenzyme A through acetyl-CoA, we released it here, we used it up here, and now we release one here. So really at the end of the cycle, there is a net one coenzyme A that is generated. So this name synthetase, which is a ligase, this name of this enzyme is actually given for the reverse reaction, where you take succinate and coenzyme A and use the energy from a GTP or ATP to form succinyl-CoA. So ligase is a class of enzymes that joins two molecules using energy from ATP or another nucleotide. So the name of this enzyme comes from the reverse direction. In the next reaction number 7, the succinate is oxidized to fumarate. Basically, one hydrogen from here and one hydrogen from here is removed, and you get this double bond here in fumarate. The oxidizing agent is FAD. It takes the two hydrogens to form FADH2. This enzyme is called succinate dehydrogenase. Once again, it's an oxidoreductase. Succinate dehydrogenase is the only enzyme of the Kreb cycle that is actually bound to the inner mitochondrial membrane. So this is actually a membrane-bound enzyme. The rest of the enzyme of the Kreb cycle are found in the matrix. So this is the only membrane-bound enzyme. And the reason is because this FADH2 will donate its hydrogens to complex number 2 of the electron transport chain. Now the electron transport chain is in the membrane, the inner mitochondrial membrane where this enzyme is. So this enzyme being very close to the electron transport chain, complex 2, will donate its hydrogens to complex 2, and we'll talk about this in the next video on the electron transport chain. And when it donates its hydrogens, the FADH2 gets reconverted back to FAD to continue reaction number 7. So FAD is covalently bound to succinate dehydrogenase. Remember, we're talking about main straight, and the idea is to take all the NADH form, this NADH and this NADH and this NADH, as well as this FADH2, and send them all to the electron transport chain oxidative phosphorylation system so that we can generate ATP. Once fumarate is formed, in reaction number 8, we have basically an addition reaction. Water is added across the double bond to form malate. The enzyme that catalyzes this reaction is fumarase or fumarate hydratase, and it belongs to that family called lyase. In the next reaction, the hydroxyl group of malate is oxidized to a ketone, and malate gets converted into oxaloacetate. The enzyme that catalyzes this reaction, number 9, is called malate dehydrogenase. It's an oxidoreductase that catalyzes redox reactions, and here NAD is the oxidizing agent that oxidizes the alcohol of malate into the ketone of oxaloacetate. So the oxaloacetate that was used in reaction number 1 has been regenerated by reaction number 9. So basically, to oxidize the acetyl-CoA into carbon dioxide, we have not used any of these intermediates. We have not used oxaloacetate or any other intermediate of the Kreb cycle, and that's why none of these intermediates of the Kreb cycle appear in the net equation for the cycle. So you can see from the net equation of the cycle, acetyl-CoA goes into the cycle, the 2 carbons of each acetyl-CoA come out as carbon dioxide, and what we produce is products like NADH, FADH2, GTP, and coenzyme A. So as far as energy is concerned, you can see that every acetyl-CoA that enters will give you 3 NADH. So for glucose, from glucose, remember we're getting 2 acetyl-CoAs, so that will give you 6 NADH. So for every 1 acetyl-CoA, you get 1 FADH2. So from 2 acetyl-CoA, you get 2 FADH2. From 1 acetyl-CoA, you get 1 GTP by substrate level phosphorylation. From 2 acetyl-CoAs, you get 2 GTPs. So when you add all this up, 6 times 2.5 ATPs from an NADH to the electron transport chain oxidative phosphorylation system, FADH2, each one gives 1.5, so 2 times 1.5, so we have 15, 3, and then 2 GTPs is the same as 2 ATPs. Add all this up, and we get 20 ATPs in the main street, in this phase of main street, 20 ATPs from the glucose, just from this Kreb cycle. So remember, each glucose gives you around 30 to 32. So Kreb cycle gives you quite a lot of glucose here, 20 out of the 30 to 32. So glucose, remember each glucose has 6 carbons, and as far as losing the carbons, we are losing 2 of the 6 carbons in the PDase reaction, and then 4 of the 6 carbons in the Kreb cycle or TCA cycle. All except 3 reactions of the Kreb cycle are reversible. So only these 3 catalyzed by citrate synthase, reaction 1, isocitrate dehydrogenase, reaction 4, and alpha ketoglutarate dehydrogenase. So these 3 reactions are the irreversible reactions under cellular conditions. And as we'll see later on the regulation, when we talk about regulation of metabolism, these are the points where the cycle is closely regulated. Some other important points to note about the Kreb cycle, as far as breaking down acetyl-CoA into carbon dioxide, it is a catabolic cycle. So we are using oxidizing agents to break down acetyl-CoA into carbon dioxide. The intermediates are not used up, so they're regenerated, so they're not used up. As you can see from the net equation, none of these intermediates, oxaloacetate, succinate, or whatever, appear in the net equation. And that's very useful. So basically the cell can send acetyl-CoA into the cycle. It doesn't matter whether this acetyl-CoA comes from glucose or whether the acetyl-CoA comes from fatty acids or amino acids. It doesn't matter. So basically the cell sends acetyl-CoA in, and out comes ATP as substrate level phosphorylation, and NADH and FADH2, which will give you more energy. So in goes acetyl-CoA and out comes a lot of energy. And all this can happen without using up any of the intermediates of the cycle. So big advantage to cells as far as energy generation. Now, besides energy generation, Krebs cycle is also very important because it connects with many biosynthesis pathways that we'll look at in the future. For now, just to give you a few examples, oxaloacetate is used to make glucose. That's by gluconeogenesis. And we'll see this becomes very, very, very important in starvation, early starvation, and even more so in late starvation. Citric, which can be converted into acetyl-CoA, and then this acetyl-CoA is used as a very important building block for fatty acids as well as for cholesterol. Other intermediates of the Krebs cycle can be used to make amino acids. So for example, oxaloacetate can be used to make certain amino acids. Alpha-ketoglutarate can be used to make certain amino acids. So the Krebs cycle intermediates are also important not only for glucose synthesis and lipid synthesis, but also for eventual protein synthesis. Succinyl-CoA, which is an intermediate of Krebs cycle, is a building block for porphyrins. So porphyrins example is the heme group. So in order to make this heme that is necessary for making things like myoglobin and hemoglobin and cytochromes, the building block for the heme group is this intermediate of Krebs cycle called succinyl-CoA. So clearly Krebs cycle intermediates are used quite a bit for biosynthesis of other molecules. Now, if the Krebs cycle intermediates are used for biosynthesis of other molecules, there is always a possibility that, let's say we're making a lot of glucose, there's always a possibility that we may not have any more oxaloacetate present anymore. And if oxaloacetate is not present, then the whole Krebs cycle is going to stop, and that is going to be very bad for the cell, because that's going to seriously affect its energy generation. So to make sure that the Krebs cycle does not stop because one or more of the intermediates is no more present, there are several reactions that actually make these Krebs cycle intermediates. These are called anaplerotic reactions. Anapleresis in Greek means filling up. So we are basically filling up the Krebs cycle to make sure that none of the intermediates disappear. Probably the most important anaplerotic reaction is this one here, where pyruvate can be converted into this intermediate, this very important intermediate oxaloacetate. And this is catalyzed by an enzyme, pyruvate carboxylase, that we'll talk about in a future video. The breakdown of certain fatty acids or chain fatty acids can also provide this intermediate succinyl-CoA, and several amino acids breakdown can provide intermediates such as oxaloacetate, fumarate, succinyl-CoA, as well as alpha-ketoglutarate. So these reactions make sure that the cycle will keep on happening all the time. So one last important point, humans cannot convert acetyl-CoA into glucose. as we talked about in the previous video. That's because the PDS reaction is irreversible and humans have no other pathway available to convert acetyl-CoA into glucose. However, bacteria, fungi, and plants do have another way to convert acetyl-CoA into, in this case, oxaloacetate, because oxaloacetate, as we shall see later under gluconeogenesis, can be made into glucose. This is called the glyoxylate cycle. So this cycle allows bacteria and yeast to actually make carbohydrates from lipids, okay, which humans cannot do. Now, clinically, this is important because they are, for example, the bacteria that causes tuberculosis, Mycobacterium tuberculosis, and there's certain yeast strains. These microorganisms can live within human cells for years, and that's how they can avoid immune destruction. So how do they survive in these cells for long time? Well, what they do is they use their lipids, okay, and they store quite a bit of lipids. They can use their lipids to make acetyl-CoA, and then use this acetyl-CoA to make glucose. So they can make carbohydrates from the glyoxylate cycle, and then they can use the Kreb cycle, to make the amino acids. Okay, so they can use the intermediates of the Kreb cycle to make their amino acids, their proteins. So in this way, they can make their carbohydrates, they can make their amino acids, they have their lipids, and so they can survive for a long time, hiding in our cells and avoiding immune destruction. So this is some of the important aspects of the Kreb cycle. In the next video, we shall discuss the last part of main street, and that's the electron transport chain coupled to oxidative phosphorylation, which we'll call ETCOP. So until the next video, thank you for watching. Have a good day, and hopefully we'll see you soon.